Hi class, and welcome to this week's podcast on non-Mendelian genetics. So what we've covered so far in the previous videos, we had Mendel, who he was. He was the father of genetics, a very important person in genetics. We had Mendelian genetics, the thought that there is only an either or trait. So it is simply dominant or simply recessive. There's not really any in between in this. Now, what we're going to learn today, not all inheritance patterns are simple to follow. So they are not all like what Mendel discovered. So we did have either or. We're going into more complex ways to inherit. So a little bit of recap again. The dominant recessive type of inheritance is the thought that it must be one of two traits. So you have the dominant trait or you have the recessive trait. Kind of like, remember our hitchhiker's thumb? Well, that was our dominant or recessive trait. The dominant being that you have the hitchhiker's thumb, the recessive being that you have a straight thumb. Today we're going to go into incomplete dominance. Hybrid organisms in incomplete dominance tend to have the trait of a blending of the two phenotypes. So, for example, with flowers, if you have a recessive trait being white in flower color and a dominant being red in flower color, we get a hybrid or a heterozygote, which is pink. So you can see the blending of the two there. And then we have codominance. Two alleles are expressed in the hybrid form. So when you see a spotted cow, you're going to see two colors. Those are both dominant forms versus having a solid color, which is only dominant or recessive. Okay, let's go on to incomplete dominance. So I know you have an understanding of what dominant and recessive is. So we're going to fill out this chart on incomplete dominance. So when you cross two snapdragon flowers, a true breeding red and a true breeding white, what genotype and phenotype do you end up with? Alright, so we have the true breeding red, and we're going to draw an R for our true breeding red, capital R. And then we have a true breeding white, which we draw a capital W for each of those. Now, as you can see, our notation here in incomplete dominance is a lot different than our notation for dominant recessive. In dominant recessive, we had capital letters and lowercase letters. In incomplete dominance, we have a capital letter for one type and a capital letter of a different letter for the other type. So we have an R and a. So with each of these, we're going to write out our genotype. Remember, genotype is the actual genes that we have. And with every single one of these, we get an RW. The RW is going to be the phenotype white. So we have the genotype red and white, or RW, and the phenotype is going to, or the phenotype of our flower is now going to be pink. So we have a pink flower with both R and W as their genotype. Now, as you can see, we have 100% that are going to be that phenotype. So, pretty important to know. Okay, let's move on and talk a little bit about this. Now, as you can see here, this is going to show you incomplete dominance and what is happening with incomplete dominance. I would really like you to put this in your notes, so pause the video, put it in your notes, and we'll go over it on Monday. Okay, so you have a white flower crossed with a red flower, and they are both true breeding flowers. As you can see, just like we just did, you get all pink flowers. Now, I don't want you to look at the notation here. The notation here is incorrect. I want you to only notice the colors on this page. So then for our second or F1 generation, remember that means filial, we cross a pink with a white. And when we cross that pink with a white, we're going to get half pink and half white. Because remember, the pink has the genotype of RW and the white has the phenotype of WW. So when we cross that, you can draw that Punnett square out in your notes. Actually, I would really like you to do that. And you will see that you will get RW and WW as the possible phenotype. So we have a 50-50 here on what we can get. Now, as you can see, if you cross two pink flowers, you are going to get a 1:2:1 ratio. It's pretty common. Remember, we talked about that in Mendelian Genetics. So we get one possible white, which is our recessive form. We have one possible red, which is our dominant form. And for our hybrids, we get pink. So that 1:2:1 ratio. Okay, I want you to go ahead and do this on your own. Pause the video, and this will be collected. So do this on a separate sheet of paper. Codominance. Codominance, we have a new notation, just like before. It's not the same notation. So you choose a letter representing the gene, and that letter can be any letter. The only letters you cannot choose are X, Y, or I, as they are designated for sex-linking genes and blood typing. So if we had a cat that we knew was either white or red or spotted due to codominance, in codominance you have two dominant genes, which are both expressed if you have the hybrid form. So we choose a capital letter. It might have to do with the cat's coat, so we have C, or you could even do C simply just for cat. Now, what we are crossing is true breeding. Remember, true breeding means they have two of the same gene. So we have a capital CW by a capital CR. You must use the superscript when doing codominance. This is what shows us that we are doing codominance versus a general dominant recessive or even incomplete dominance. So we use this different notation. It helps us when we're just looking at a problem. Okay, our first problem in codominance. For shorthorn cattle, the mating of a red bull and a white cow produces a calf that is described as roan. Roan is a type of cow that is red and white spotted. You've probably seen it and didn't even know. Now, roan results from intermingled red and white hairs. Many matings between roan bulls and roan cows produce the cattle in the following ratio: 1 red, 2 roan, and 1 white. Now, what I want you to do, go ahead and do this on your sheet that you're going to turn in. I want you to do a cross between two roan cows. So a roan bull and a roan cow, and show me that you will get the 1 red, 2 roan, and 1 white ratio. Moving on, codominance is how we do our blood typing. It's very important. Blood typing, just one of your genes that you have. Now, you might not know exactly what each blood type means. That's okay. We're going to go over it. But for now, I would like you to put this table into your notes. It's very important. You need to memorize this table. This table will come up in other activities in class and just other activities in general. Okay, so blood types are inherited by codominance. They're indicated by the symbol I. You always have to use I. This is not one that you get to choose. Remember when I was talking about what you can and cannot use, I was already taken, and that is due to the fact that we use it for blood typing. Now you can have capital IA, capital IB, or a small letter I. Now if you look at the chart below, you can see why we have this. The capital IA is a dominant form. A and B are both dominant. Now you can get the recessive form, you can have IA little I, which is still going to give you the blood type O. Same with B. You can have IB and little I, which is still going to give you the blood type B. And that little I is known as a recessive form, which is O. And you've probably heard of people having O blood. Now, if you get both IA and IB, you have the form AB. And two little i's are going to be the form O. So what I want you to do, this is another problem for your sheet to turn in. You're going to pretend that Brad Pitt is homozygous for the type B allele. Remember, homozygous is two of the same gene. And Angelina Jolie is type O. So what are all the possible blood types of their baby? Let me know. And I want you to do this on that sheet. Mrs. Essie is type A and Mr. Essie is type O. They have 3 children named Matthew, Mark, and Luke. Luke is adopted. Mark is type O. Matthew is type A. And Luke is type AB. Please answer A, B, and C and draw the Punnett squares that go along with them. Okay, hold on. As you can see on the blood types here, we have type A, B, AB, and O. Type A has the antigen A on the cell, where it has the antibody in your plasma B. So see this right here? That is your antibody. That is why we can only receive certain types of blood if you are type A. Type A can only receive from type A and O. Now in type B, we have the type B antigen on the cell, and in the plasma we have the antibody A, and type B can only receive type B or O. Now we have type AB. You see it has both types of antigen on the cell and no antibodies. Type AB is the universal receiver. He can receive blood from A, B, or O. And then type O is kind of just the exact opposite. He has nothing on the outside of his cell. He has no antigens, but he has both A and B antibodies. So those antibodies are the same as you have in type B and A, which makes him the universal donor. Anyone can accept type O blood. Okay, let's move on to sex-linked traits. Sex-linked traits are traits that travel on the sex chromosome X. Now, as you can see right here, this X is a lot larger than this Y. This is due to the fact that X has, everyone has an X, and X has many, many different types of genes, where Y, not everyone has a Y, only boys have a Y. So it has a sex-determining gene. So if you do not have a Y, you actually do not have the sex-determining gene. And what our bodies default to then is female. So males have an X and a Y, so they can be determined to be male, and that gives them those secondary sex characteristics. Where females have 2 X chromosomes, while we do not have the sex-determining gene, our body defaults to female. Okay, so our sex-linked examples. Eye color in fruit flies is sex-linked. It is carried on the X chromosome. And what we're going to do is we're going to cross a red-eyed male by a white-eyed female. Now notice the notation again. You have a capital R, Y, nothing on the Y. I repeat, there is nothing on the Y. This is due to the fact that we have such a large X chromosome, and that's crossed by a white-eyed female. Capital X little r, capital X little r. Go ahead and do that in your notes form. Okay, and now you can see, based on what we have, in sex-linked traits like hemophilia, women are carriers and men have the phenotype more often. That's because women have 2 X's and men cannot carry things on their Y. So they only have one chance to either have something or not have something. So as you can see here, Queen Victoria was a carrier, so was Alice, so was Alexander, and finally Alexis had the disorder. Alexis was a male.